Hi, I'm Shannon Wyman, Scientific Communications Manager at Keystone Symposia, and I'm here with Dr. Malene Hansen, co-organizer of the Keystone Symposia meeting on Intra and Intercellular Mechanisms of Aging here in Vancouver. Thanks for joining us today.
Absolutely.
So what's come out of the meeting? What are the new and exciting advances and directions in this field?
Yeah, I think it's been a phenomenal meeting, actually. The design of the meeting was to highlight that not only are there intracellular mechanisms in terms of decline for aging, but it's clear over more recent times that there is communications between tissues. And so learning more about the intertissue communications and that, your cell is not alone, but will talk to other cells in an organismal way to affect both aging and age-related diseases were sort of the topic and has been really standing out in many, many, many ways for understanding circuits of signaling and also how those signals would be sort of communicated between the different tissues in an organismal kind of fashion. So yeah, I think the many different model organisms, the many different approaches that people have taken to really start and to decipher this sort of extra level of complexity that there is to an already very complex process like aging is really starting to unravel. And yeah, there were many really good examples to that.
And what kinds of signals are coming out? What types of molecules or proteins or?
Yeah, so naturally a lot of the signal revolves around the brain or neurons producing signals to distal tissues, but also now even more, I would say, starting to be sort of like looked at it the backwards way, like other tissues signaling back to the brain. So that sort of become the next big question. What are the molecules that actually transfer this kind of information? And so a lot of neuropeptides are naturally being put forward. I think they need to be investigated in a lot more detail, but even just analyses of blood for different factors that could have such communication facilitating, aspects to them, I think is really, truly exciting. Yeah, so I think the molecules is going to be diverse. There's going to be many of them, each of which would present its own opportunity for maybe in the long goal, like with an intervention of some kind, right? Like associated with the diseases that this particular tissue, for instance, would experience over time. Mm-hmm. So it's very exciting. But there's a lot more research, obviously, still to be done in order to figure out exactly how to manipulate in a very specific way. Mm-hmm. Yeah.
So what are the open questions then still, and what are the directions that the field is going to be looking into?
Yeah, so I think we need more of the basic understanding. I think the field has for many decades concentrated for good reasons on the intracellular mechanisms to see what happens during decline. And so this new level or complementing level of understanding how do something that is dysregulated on a single cell level now talk to other tissues, I think is where the field is, but definitely also still is going in terms of really understanding the complexity, and that will take a long time. But the good news is also, and that was represented well also at the conference, that there are new tools and new techniques that will allow us to really approach those questions. It's a very exciting time, just because technically we're also in a much stronger situation, right, to kind of approach some of those problems.
What are some of those tools, and what do they reveal that we weren't able to look at before?
I think a lot of it is sort of many of the approaches that are used traditionally would be genetic approaches to try and ask what happens when you modulate a gene. And so with new gene editing techniques like CRISPR is truly revolutionizing how expeditedly we can sort of go through many of these mechanisms. Cell biology-wise, there are ways to now look at the tissue in a much, much more high-resolve way with like mass spec approaches on a tissue. So I think the complementation of these genetic approaches with biochemical approaches and cell biological approaches, it's all definitely coming forward in a way that now in this case also is combined with sort of the context of aging, right? So one extra aspect to sort of add-on. So these projects need to be planned in a very careful way, right? Because once you then start a full lifespan analysis of a mouse, for instance, right, these are years of work. So it's very important to kind of learn about these techniques at a place like a Keystone conference to really bring that back and think about how can we do our projects better. Yeah.
How did you see people interacting at the meeting, bringing in these different approaches, different techniques, and different directions and perspectives of a study to integrate these ideas from the inter to the intra?
Yeah, so that's probably one of the most important aspects of a conference. It's people come together and exchange not only sort of specific knowledge about each and an individual projects, but like how can I maybe use some of these techniques in my own projects. And so I know from having talked to many of the conference goers, right, that this is sort of the high of a conference, like you leave with, because you now have access to a new reagent, or you've discussed a new approach or a new question that can maybe be pursued in a new collaboration. So that's always exciting, right, that there is this exchange of information that then fosters all these new ideas. So I do feel that there's been a lot of conversations to that end, and we're excited to pursue new ideas as well.
Mm-hmm. Great. And then looking into the signaling as well as the mechanisms that go on inside the cell, what are the implications for either preventing or treating aging and or aging-related diseases?
Right. Yeah, so that's obviously the ultimate kind of goal, right? That we study, or I speak for myself at least, I study aging to understand the mechanisms that could then be applicable not only for, let's say, one age-related diseases, but many in that regard. So I think the conference was a good mix of having many of these basic research observations that could be sort of the stepping stone for the next big thing, even though it will take a while to develop. But there were also projects that are, let's say, more direct in the sense of, you can exercise and there would be real good benefits to something like that, right? Like that's something you can do immediately, more or less. Mm-hmm. New drugs and other things like that are, also being discussed. And so it takes a long while, right, to kind of test it all through the different systems. But I think there's definitely several links and new ideas that are going to be exciting to follow in the future.
So what are some of the new targets, whether those are molecular or just kind of thematic, that are emerging?
I think sort of the standing information has been, and we have looked for many years for new molecules, biomolecules, or genes and pathways that could be targeted in many ways. I think we understand more about those genes and also how complex it is that when you sort of modulate one type of process in the cell, I mean, you may also affect other processes of the cell. So that's both, I guess, higher complexity, but also an opportunity to better integrate and learn how to do that. So new targets, I think there were some new ideas that have come forward that people can learn and obviously also think about in a conservation manner, right? If it's an observation done in a simple organism like C. elegans or a nematode like C. elegans, right? Like we would want to look at them in like a fly or a mouse, and so meeting other people that study things in other organisms is something that provides an opportunity as well. Mm-hmm. So many of the early observations have come from simple organisms like C. elegans, but it would be really important to now test them in other systems. Mm-hmm. Yeah.
And for you, what was the most exciting or interesting or fun part of the meeting?
I thought, it was a great meeting. There are many scientific highlights. It's also a great opportunity to meet friends and make new friends. I was excited to see how many junior people were here and also the quality of their presentations. We had many of them chair sessions, and you can tell that they have a lot of excitement for the field, and I think that was sort of a highlight to know that there's very likely a bright future for aging research in that regard as well.
Great. Touching on the junior researchers, I also wanted to discuss a little bit the career roundtable workshop and how that came to be and how it ended up materializing.
Yeah, so I think mentoring is an important component of doing science as well, in terms of helping people move forward their careers. There's a lot of advice that you can take. You don't have to necessarily learn all of these lessons. So yeah, so that was an opportunity, I think, also at an international meeting where you definitely get people from many different places and different career tracks to come together and just hear what are some of these thoughts and ideas and questions and concerns. So putting it together this time, we tried out like a speed dating format, or more like a roundtable discussion format that also provided a better networking opportunity to meet more of the senior people at the meeting. And so there were many concepts that came forward that I think are important to discuss. For instance, for a junior faculty, like how do I decide what to take with me from my old lab? You know, how do you navigate many of those questions that can be, quite important when you sort of set off. And so I thought it was fabulous. There's a lot of great conversations, and they continued much more even after we closed our little roundabouts and the tables, yeah.
And what career perspectives were brought into the discussion then?
Yeah, so we had, and I assume you can do more, we had an editor table to discuss also journals in general. We had a table for NIH and government to discuss more like the funding avenues for and also career opportunities in there. And then we had a table on basically junior PIs and senior PI in academia, and then a table on industry and career tracks and ways to sort of succeed within that. So yeah, there's good discussions amongst all of those for just learning simple things. Like you can call an NIH officer; they're there to help you. They don't just sit in a closed house. Or so there were many kind of exciting, maybe obvious comments, but also I think some very important ones and lessons for everybody to take from there.
Mm-hmm. Great. Was there anything else you think people should know about the meeting or the field in general?
I think the aging field is an exciting field. It has its own complexity, but because aging is a complex process, it's really helped by having many different people from many different areas. And so the conference itself become broad, but the biology that comes forward is really exciting and important in that regard. So I think it highlighted very well that aging research is complex, but is well studied by doing it in many different ways.
Mm-hmm. Well, thanks so much for your time, and enjoy the rest of the meeting.
Thank you very much.
Talking
Thank you very much.